Good morning students, welcome to online class Padmavathi Junior College, Narasannapeta. I am A.bhaskar, Botany faculty. Today we discuss about the topic plant physiology and microbiology. Various kinds of chapters are there in our textbook. 6 units they are present in our textbook. First one is plant physiology and biotechnology. Next microbiology, next వచ్చేసరికి genetics, molecular basis of inheritance and plant microbes and human welfare. Today we discuss about that fourth unit, molecular basis of inheritance. Molecular basis of inheritance. We obtained 8 marks, one 4 marks question and two 2 marks questions from this chapter. This is very important, 4 marks questions. Here we have discussed about that transformation. Transformation. What is meant by transformation? Is defined as uptake of NAC DNA fragments NAC DNA fragments of the surrounding environments. Surrounding environment. So this is transformation. Uptake of NAC DNA fragments of the surrounding environment and the expression of the genetic information. Information in the recipient cell. This is transformation. Okay, transformation, which means it is defined as uptake of NAC DNA fragments of the surrounding environment and the expression of the genetic information in the recipient cell. So the genetic transformation, it was proposed by Frederick Griffith in 1928. Frederick Griffith he performed the experiment on bacterial transformation. Frederick Griffith performed the experiment on bacterial transcription. Bacterial transcription. Sorry. Bacterial transformation, not transcription, transformation. Bacterial transformation with Streptococcus pneumonia bacteria. With Streptococcus pneumonia bacteria. Streptococcus. Streptococcus pneumonia. Streptococcus pneumonia bacteria. So, and we choose that host organism is mice. Host organism is mice. Mice means rat. Here, the Streptococcus pneumonia bacteria is causing the disease pneumonia. It is causing the disease pneumonia. Okay. So now we are going to that experiment. First of all, Frederick Griffith grow the bacteria in the staining medium. This is the staining medium. Frederick Griffith grew the bacteria in staining medium like this. Here, the bacteria forms their colonies. The bacteria forms their colonies. Here, cocci, which means circular bacteria. Cocci means circular bacteria. Okay. Frederick Griffith grew the bacteria in staining medium. Streptococcus pneumonia in staining medium. And he observed two strains. Here, two strains are there. Two colonies are there. One forming smooth colonies. Another one forming rough colonies. Here, smooth colonies. Frederick Griffith observed two strains. Here, one forming slime sheath with polysaccharide. One forming with slime sheath. Okay, here two stainings are there. One stain forms slime sheath. Slime sheath with polysaccharide. So this type of staining is known as S type. So, okay. Another one forming it is rough stains. Another one is forming rough stains. Why? Because there is no polysaccharide sheath around it. So it is known as rough type, R type. Here, smooth strain. This is rough strain. Rough strain. Okay. S type and hard. Here S type. This is virulent. Virulent. Which means this is caused to the diseases. This is caused to disease. So this is virulent. R type is non-virulent. 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 Okay. Now we are going to another step. Frederick Griffith. How do you know about that? He chose the host organism mice, which means rat. First of all, Frederick Griffith, he separate S type, S straining from the medium, and it injected into the mice body. This is the mice. When the S strain cells, S strain bacterial cells are injected into the mice body, a rat body. Here, the mice is died causing pneumonia. It is died. Mice is died. Died mice. Okay. First of all, S training. When S strains injected into the mice, mice is died. Causing the disease pneumonia. Next one. The next step, when R type or straining is injected another mice. This is the R colony or straining colonies. Okay. Or straining injected into the mice, healthy mice. Mice is not did not cause to the disease. Here disease is not appeared. So mice is survived. Is it clear? Next, another step. S straining bacteria is there. S strain. S strains. Then Frederick Griffith isolates S strains from the died mice. And heat it. Okay. S strains which are present in the died mice, they are isolated from the died mice and heat them. After that, the bacterial cells they are died. Here, heat died bacterial cells. S strain especially. Okay. S strains. Heat died S strains. Another step, Frederick Griffith. When heat died S strain bacterial cells, they are injected into the mice, another mice. So this is heat died S strain bacterial cells, Streptococcus bacteria. They are injected into the healthy mice, the mice body. Here, the disease pneumonia is not appear. Here, the disease pneumonia did not appear. Why? Because here bacterial cells, they all died. Now we are going to another step. Frederick Griffith. When heat died S strain bacterial cells were mixed with R strain bacterial cells. R strain bacterial cells, they are injected into the another mice. I understand all of you. Heat died S strain bacterial cells are mixed with R type. They were injected into mice. S strain and R strain. Okay, S strain and R strains, they are injected into the another mice. Okay, both S and R type, they were injected into the mice. Here the mice is died. It is died because causing pneumonia. Causing pneumonia. Like this. So what will happens is here S type and R type bacterial cells, they were injected into the mice. Mice is caused to disease pneumonia and died. Okay. So what happening is there here the polysaccharide sheath which is around to the S type, they are transferred to R type. R type. Why? Because here bacterial cells are died. So heat died S strains are there. So the polysaccharide sheath, they transfer R type. So here the bacterial cells, they are acts virulent. So they are changed into S type. R type changed into S type. So they are caused to the pneumonia disease and rat is died. So finally, Frederick Griffith, he concluded that R strain R strains had some have been have been transferred by S strains. Okay. Finally, it decided that transformation, the genetic material which is transferred from one place to another place. Okay. Genetic material is transformed. Okay. Already we are defined that uptake of naked DNA fragments of the surrounding environment, which means here S strain to R strain. S strain to R strain. This is the transformation. This is very important. 4 marks question. Okay, I say once. Transformation. Transformation means uptake of naked DNA fragments of the surrounding environment and the expression that genetic information in the recipient cell. Okay, in 1928, Frederick Griffith performed the experiment on bacterial transformation with Streptococcus pneumonia. Streptococcus pneumonia is causing the disease pneumonia. Okay. Next. Now we are going to the experiment transformation. Here, Frederick Griffith grew the bacterial cells in straining medium. Here, he observed 2 strains, one forming smooth colonies, another forming rough colonies. Here, smooth colonies are virulent, which means they are caused to the disease. After they are non-virulent. Just few minutes. Or virulent. Okay. Now, S type. S strains, they are injected into the mice body. Mice is caused to the disease pneumonia and die. Next, another one. R strains, they are also injected into the another mice. Mice is not died because it is not caused the disease pneumonia. Next, the S strain cells, they are isolated from the died mice and heat them. Here bacterial cells are died. Heat died strains are mixed with R strains. They are injected into the another mice. The mice is caused to the disease pneumonia and died. So what will happen is there. Here R strains are somehow being transferred by S strains. What is that? Genetic material. What is the genetic material here? Polysaccharide seed. Like this. Polysaccharide seed. Okay. Thank you all of you.